So before proceeding with Dr. Amine Kamenand, I would like to actually briefly introduce him. He's a professor of bioengineering at McGill University, Montreal, Canada, and holds a Canada Research Chair in bioprocessing of viral vectors and vaccines. He was until end of 2013 the head of process development section of the Human Health Therapeutics portfolio and lead of the bioprocessing and manufacturing of the vaccine program at the National Research Council of Canada. So he received a PhD in chemical engineering from Paris and a PhD in biochemical engineering from École Polytechnique de Montréal. So before joining the NRC in 1991, he was a professor of mass transport phenomena and reactor design. His current research activities focus on developing enabling technologies for manufacturing of biologics. He has been involved in scale-up of industrial processes, rational media design, metabolic engineering, process control, and monitoring of high cell density and high yield productions of recombinant proteins, viral vectors, and vaccines. He published more than 150 papers in refereed international journals and also acts as consultant for many national and international organizations. So, sir, we are really lucky and fortunate to have you here today, sir, with your kind of experience and expertise. Even if we can imbibe 1% of what you are doing, that will be a great opportunity for us, sir. So I request you to kind of talk.
Thank you for the kind introduction. Actually, it was quite extensive, and I will try to give example actually of how these really apply. So I'm trying to share my screen. So you have to allow me sharing the screen.
I think I have, because
how's disable attendee screen sharing? This is what I'm getting as message.
Just give me 1 second. Every time
it's you have to go to share screen, and you have an option, advanced option there normally, and allow sharing screen for all.
It says screen sharing is on, so that's why it's, because last thing we didn't have this meeting settings. Yeah, are you able to see that now?
Share screen, yes, it's working. Yeah. So I will go with my presentation PowerPoint share and put it full screen, and let me know what you see in your screen. Is it the presenter view?
Yes, sir, we're able to see your vector vaccines.
Okay, but is it the presenter view, or is it full screen?
It is not. It is presenter view, sir.
Okay. Swap display. Is this better?
Yes, sir. This is better, sir.
Okay, this is what I was looking for. Actually, these are some tricks with with Zoom. So I understand that I have 50 minutes to go over, and
is it? Yes, 1 hour. Yeah, 1 hour.
Okay, so so it's a pleasure actually to contribute actually, and I felt that it's very appropriate to cover one topic that is obviously of importance these days, and it's something. I just finished a course, in fact, of 50 hours just on bioprocessing of vaccines. So it's and I get a lot of excitement with the participant, just because it's extremely important to realize actually that we are depending on this type of product, but you understood that our perspective is with regard to bioprocessing. So what I will try to do today is give you an example. In fact, we cannot cover it all, but an example that turned out to be one of those approach that might be effective in time of pandemic situation, of rapid response. And you might be surprised actually that, I mean, over the last 10, 15 years, people were, I mean, the one that were definitely in the field, following very closely, I mean, the problematic of potential pandemics, and the expectation was more with something very similar to flu, but turned out that it's slightly different, but we are still with the same pattern. And the proposal I have here is to focus on one viral vector that in fact has been originally developed for gene therapy, but turned out that in that space it wasn't that successful, and have been in a way recycled to be used as a vector for delivering an antigen. This is the notion that I'm developing. In fact, instead, for any infectious disease, the identification of the dominant antigen or the critical antigen is the first step, and you design by having a vector that is essentially a virus, but safer virus to be used to expose or to express the antigen of interest. And here, so the example is with the adenovirus, and the adenovirus, we started working with the adenovirus toward rabies control. And as you will see, this is an interesting story that also will link something extremely important in the area of vaccine at large, which is the so-called principle of one word, one health. And there is a connection between what's happening in the animal space, veterinary vaccination versus human protection. I will give the example on the Ebola vaccine, and I will give another example that is currently we are working on Newcastle disease virus, and obviously you might guess that we are also working on the SARS-CoV-2 as a potential vaccine. I will cover the vesicular stomatitis virus that was one of the most successful, in fact, for the Ebola vaccination, and it's currently an approved vaccine in North America. And I will briefly just indicate on other vector for gene delivery. The pox is well known, and eventually the baculovirus that play a role in some of the vaccine. I forget to say, if anything is becoming totally unclear, just stop me and let me know, and I will be happy to respond. So before getting further, I would like just to summarize that when we refer to viruses, we cover the kind of quite large scope of application. And this cover, and I mean, we spend quite significant time in developing vector for gene therapy, we still do. And in this case, we will be considering the so-called replicative defective vector. You want the vector just to deliver, and definitely not to replicate. So you manage the modification of the vector in such a way that it's getting inside the cell, delivering the genomic payload, but do not replicate, because you don't want to create any additional harm. The second category that is definitely emerging over the last, I would say, easily 20 years is the so-called oncolytic therapy. Here you will be using a virus in a way that it will be specifically infecting tumor cells and replicating specifically in tumor cells, allowing therefore, I mean, the destruction, if we may say so, of the tumor cells, and the specificity is such that it's not replicating in normal cells. The traditional way to look at vaccine is essentially taking the infectious virus or a component, which is essentially the antigen. And in that case, we will be delivering the antigen, and this is the purpose of the talk today, delivering an antigen or even just displaying it in the surface, as we'll be looking at. A field that emerged is the so-called virus-like particle, and in fact there is example of vaccines here that have been took some time to develop, to design, to develop, and to manufacture. The good example is the HPV vaccine. In fact, it's the so-called viral-like particle. The key point here is you take the external element of the virus, essentially capsids, and manage to get a structure that is very like the virus, but without any genomic material. So from that aspect, it's considered as safe as compared to the totally normal vaccine that is what is has been considered, which is probably 50% of the vaccine that are in current use, and we don't have in reality that many vaccines. I mean, it's probably something in the range of 30 vaccine. Product is different because there is a mix of a certain number of vaccine, and here we will fall in the area of just taking the infectious virus. In the case of the COVID-19, this is there is a clinical trial targeted with inactivated vaccine. The challenge being in this case, it's the whole virus. Because for the COVID-19, the critical point is that this is a level 3 agent, and it has to be manufactured at within level 3 capacity. Attenuated is a live virus. However, it has been attenuated by multiple passages in such a way that it's going to be an effective vaccine, but without with higher safety. So, if this is the scope of the area we will be covering, with essentially a focus on what's in the middle, the vector vaccine. So far, so good. Any critical question?
They will place the questions at the end of the seminar in a chat box and go with the questions.
That's fine. That's fine. But my point is that don't hesitate if there is anything of urgently needed to clarify. Let me know. So when we refer to vector vaccine, this is already a reality. Actually, the naming might be different, but as soon as you take a virus and equip it or modify it recombinantly, using another antigen, we are dealing with the concept of vector vaccine. And when I'm saying it's a reality, there is already vaccine on the market. From Sanofi Pasteur, for example, the vaccine against Japanese encephalitis is using as a vector the attenuated yellow fever vector 17D, and replacing essentially by the dominant antigen of the Japanese encephalitis. It's exactly the same approach in the tetravalent dengue vaccine. So Sanofi has a more different reference, referring to chimeric vax dengue virus. But the concept is already in place after years and years of safety use of the yellow fever vaccine. They decided to use that strain as the vector to expose or deliver the other antigens. So when we cover what's ongoing in the space of development, it's very significant, all the activity, and I reviewed this in 2014. In fact, it's just in the area of influenza vaccine. I will not spend too much time on this, but it's one of the challenges, actually. You know that for influenza, we have to make the vaccine every year, and that's not trivial. So for this purpose, alternative to traditional vaccine that is made in eggs just by replicating the circulating strain, there is a lot of research work ongoing with vectoring the influenza dominant antigen that is known to be the hemagglutinin, and this use the adeno-associated virus has been tried. This is a tiny virus, 20 nanometer, alpha virus, baculovirus. This is a system for recombinant protein expression, but turned to be also a very good system for delivery, even in mammalian cells, although the virus is insect cells. Newcastle disease virus, I will touch on it, parainfluenza virus, pox virus are considered vector, and vesicular stomatitis virus, and I will give more detail on the adenovirus and cover briefly the pox virus and the VSV, vesicular stomatitis virus. So when we refer to the adenovirus vector vaccine, this is where we start, actually. And because of the extensive work that has been done in the area of gene therapy originally, trying to use the adenovirus to deliver a transgene for correction, for essentially a well-identified genetic disease, a lot of progress has been made in that space by knowing extremely well the virus with the fiber exon, and this is an extremely well-known virus. It's classified level 2, so just realize that any of us do have infection with this serotype, the serotype 5 that we'll be considering the human one. It's mild cold type of infection, so that's the reason it's considered as safe for delivering or for vaccination. So it's well characterized. It has a broad tropism. This is what we want. When we want to target different tissue, we would like to have broad tropism, and this is due to the a type of fiber that allow the entries a number of cells, low pathogenicity, we just touch on it, infection through respiratory tract. And you might guess that, and we will get back to, there is already clinical trial with the COVID-19 as using the adenovirus. So briefly on the genomic part, the whole genome is quite large, 38 kb. And the work that has been done is, when we refer to defective adenovirus, there is essential region, essentially the E1 region that play a critical role in the transformation of cells, has been removed. You make a little bit more space by removing the A3 that is non-essential also for the replication. When we refer to the first generation defective adenovirus, this is what we mean. It's defective in the E1 essentially, and eventually the E3. Further work has been done to make the virus with less and less elements, which require complementation. Ultimately, we worked on this system that is so-called third generation or gutless adenovirus. You remove all the genomic element that code for the adenovirus protein. And this is a huge capacity for delivering transgene, in fact. But the downside is that you have to produce this vector, and for the purpose of producing, you need complementation. In the case of the first generation, that's straightforward because we have a cell line that does that, which is the HK293, that has the complementation with the E1. Very briefly, and this is a little bit odd, it can be easily updated, that the adenovirus quite successful in a number of clinical trials as adenovector vaccine, phase 1, 2, and 2, 3. In infectious disease, HIV, it has been used extensively. Unfortunately, it didn't make a significant difference, and many of the clinical trials up to thousands of people have been vaccinated for that purpose. The one was referred to as the STEP trial, and Merck and Co was involved heavily in that trial. There is malaria, Hep C, Ebola, I will tell you the story, tuberculosis, we worked on this aspect too, rotavirus is considered, but another area that is of active work in the clinic is the use of the adenovirus as a therapeutic cancer vaccine. And this is what I was referring to, equipping the virus in such a way that it will be replicating essentially in tumor cells and not replicating in normal cells. So in a nutshell, this is what we do in when we produce. We need a complementing cell line, and the usual complementing cell line is the so-called, it's a human cell line that had been isolated in the 70s and transformed essentially by sheer DNA of adenovirus, and that's the reason it acquired the E1 region. It has been demonstrated that after that the HK293 do have the E1 region, and that's what provokes the transformation, making this as a continuous cell line. And this is the preferred host, otherwise you will have to make a specific host that will complement the defective adenovirus when it's missing the AE1 region. For the purpose of using this defective virus will not replicate because it will be lacking the E1 region. I hope this clarified, but in any case, it's not essential for the rest of the talk. When we say helper free replicative defective, we refer to the fact that we are using the first generation adenovirus, and in most of the clinical trials, this is the one that has been used, with one exception I will tell you a little bit thereafter. The story of the adenovirus was activated by an event that happened in clinical trial with regard to gene therapy application, and it is in the memories. Well, it's 20 years ago actually. There has been one single death that moved the whole field, and that happened with adenovirus delivery. To a boy of 18 years old. It's referred to as the Jesse Kelsinger case, and that had major impact on the field of gene therapy for a while. And currently the gene therapy is recovering overall, but it raised one critical question: quantification, quantification, quantification. And as part of a number of committees with the FDA, etc., people realized that not sufficient tools were in place actually to quantify appropriately what was injected to human in clinical trial. At that time, we were relying just on UV detection, on sample preparation, that who knows the purity, etc. I'm over exaggerating, but not that far at that time. I'm referring to a little bit more than 20 years ago. So there has been a push of a focus on looking more carefully to means of quantification, and this is a key message to anyone working in this field, bioprocess development, and this is what we can bring as significant, actually, the quantitative aspect. And in most of the case, I mean, we spent a lot of time developing the analytical tool in order to support what we claim being an improvement in a process. And this is multidimensional, in fact. No need to go in a lot of detail, but for viral vector, for vaccine, we need a large set of analytical tool for essentially release, because safety is paramount, and we have to determine essentially what we are loading to the patient at the total quantity, but also what is truly functional there, and that's lead to the use of a number of assays. So this is about the story that took part at that time. It was in the 2000s, actually. There has been a consortium of all the people working in that space on the adenovirus that get together and said, what we need is a reference material. And surprisingly, that was induced by a fatal event that let's put together our efforts, make sure that we are using the appropriate method, but to test this method, let's make reference material. And that was a good example in the field. In fact, after that, there has been a number of initiatives. Last 2 years ago, we participated to an initiative for the lentivirus as reference material, and that was led by WHO. The material now, reference material, is available at ATCC. Anyone can buy it with some restriction. You cannot buy more than a number of doses per vial per year, and use it as a reference material in your lab. So what goes with the, moving a little bit on the process aspect, what is the challenge actually when we work in this field? And this is kind of generic, I would say. We work with cell lines that are continuous. Our platform is the HK293, and over the years, in fact, we contributed significantly. The main contribution was to adapt this to suspension. I'm an engineer, and my approach is to make it scalable, because we want to be able to scale up, and that is far more easier when you work with cell line in suspension. This shows just the different approach that you can reach by fed batch operation up to 18 million cells per ml. And in this space, this is quite significant. In fact, in most of the lab, cells are grown at 1-2 million cells per ml, depending on the quality of the medium that is used. I'm not referring to the CHO cells that benefited from 30 years of active development, where now everyone can grow them up to 60 million cells per ml, but most of the other cell line, we are in the range of, say, 1 to 6-7 million cells per ml. Good medium with the HK293 get you up to 10 million cells per ml in a batch mode. So this is what we face, and this is also generic. What I want you to focus on is that most of the medium that we acquire when we do cell culture are developed based on the growth of the cells. When we deal with production of viruses, which is through an infection, there is a second phase where the metabolic requirements are quite different. And in that case, you need to provide, or alleviate the limitation. And surprisingly, companies were not interested in that space for a while. They are becoming interested. So we had to deal with something that was problematic, which we refer to. And I'm saying that this is kind of generic. We find it with most of the viruses that we worked with. The first time we identified it with the baculovirus system, but here, this is the observation with the adenovirus. You infect at 1 million cells per ml, and this is what you get. I'm reporting total particle per ml, 510 to the 9. You infected 2 million cells per ml, and the expectation is that you will double the production, because this volumic production. No, you will decrease. Further at 3 million, you will even more decrease. What's happening here, we are hitting the limitation um, we are losing the specific productivity. Each cell will produce less. So that's one of the problematic as a process developer engineer over many years, we had to address actually, because it wasn't addressed by the medium manufacturer. So that went through extensive metabolic analysis using quite advanced tool, NMR, but clearly looking at what's happening during the growth and what's happening during the infection production phase. The most important for us was the production phase. And this was also the occasion to identify critical parameters that affect or impact the yield of production, something as osmolarity that we tend to take as granted. And for the same reason that the medium were developed to be efficient for cell growth, it turned out that we were not under optimal condition with regard to the osmolarity in the phase of a production. So based on work we have done, I think that this has been patented, but because it has some value actually, that I think the table shows a better summary just on this effect, and this is one effect among many, many others. We get the best specific production in terms of VP per cell, 21,000, was 22,000. In what we identified as an optimal condition with osmolarity growth of media. Sorry, there is a mistake here. This is the osmolarity for the growth media, and this is the osmolarity for the production media. Okay, so we had to have a shift between the growth phase and the production phase to get the optimal. And you see that it can be dramatic if you are in not optimal condition. We drop by essentially 20 times here by twofold, but here dramatically. So these kind of parameters are the one that we look at when we actively do process development, and ultimately this led to restoring essentially that critical limitation that we were observing. This is not looked at when you work in a tea flask, in even a shake flask, it's not significant, but if you have to run this at 500 liter scale, this is becoming quite significant and critical, and that's led, this summarizes years, in fact, of continuous improvement, and very few can work with this yield and at this cell density. So we still believe that there is a lot of work there to be completed because we haven't solved all the nutrient limitation and inhibition of the cell density and cell production. We resort to the different mode of operation, batch, fed batch, and eventually perfusion, but realize here we're dealing with a transient system. The cells are infected, and ultimately the window of operation is limited in time, so you cannot resort to extensive long-term perfusion, and we still believe that there is yet optimization to be done. I will share with you one of the primary examples that we worked on quite a long time ago, and before getting beyond this product is now had a commercial license in Canada. I started working when I was at the National Research Council of Canada with a very small company called Artemis Technology in Guelph, and they are still active. And the fact is that we have problem of rabies in Canada, but with wild animals. Similarly, the US do have significant problem, and this is the problematic. Control of rabies in wildlife remain an important challenge. And the starting point is, how can we use this adenovirus vector to deliver efficiently to wild animal vaccine against the rabies? Making the story short, these turned out to be quite successful in the preliminary assessments in the field. This is showing the main user was in Ontario, in national parks, and but very rapidly large quantity of vaccine were required for field trial to assess efficacy. And in principle, and that was my point in bringing the One World, One Health. In fact, to protect human against rabies, we go at the upstream possible. We know that the rabies are associated with bats, as it is the case with the COVID-19. The transmission agent thereafter are wild animal, skunk, raccoons, fox. And if you vaccinate at that level, and the vaccination, in fact, inside, you see my arrow, by the way? Inside, these are baits, in fact, inside which there is the adenovirus with the expressing the rabies glycoprotein. And because you are addressing very large area, it's spread, distributed by plane. This is the baiting process that is used with a kind of automatic distribution in areas, and there is identification that this led to eradication, sterilization of a number of animals that were infected with rabies. Since then, so that went very, very efficiently. Our job was to make this cost effective and scale it up. This is a 500-liter scale, actually, that is used at the National Research Council of Canada, that at that time was supporting the company to manufacture their large quantity of vaccine against rabies. Millions, I think at this time it's more than 100 million baits has been generated. At the same point, this vaccine is an advanced vaccine, and I had a lot of requests actually to support project to use it for human application. And this is the relation based on exactly the same technology. Instead of, I had to say that in the case of rabies and animal vaccination, we were using the replicating form of the adenovirus. When we moved to using human, we used the defective form. So we started the work on other antigens such as tuberculosis, and that led to some success. Phase 1 clinical trial took place. And that was the beginning of a story. The person that we collaborated with at that time was working in Canada at Sanofi Pasteur, but decided to move to China. And he licensed the process, the cell line, and created a huge capacity in China. Since then, the success was just incredible on exactly the same technology. This is the vaccine that went for phase 3 clinical trial in Africa, in West Africa, and has been approved within China. China approves domestic Ebola vaccine developed from recent outbreak, and that was as early as 2017. For China, they do have their vaccine, and it's an adenovirus base that has been approved prior to the one that was in development by GSK and Merck. And I will also refer to that. So, not surprisingly, in fact, the same company is listed as the top one in the I have it here, WHO draft landscape of COVID-19 candidate vaccine. Currently, on June 9th, there is 10 candidate vaccine in clinical trial. And guess, adenovirus type 5, CanSino Biologic, this is the company that moved in a very agile way. Things started in China. Currently, there is an agreement with the National Research Council of Canada to carry on a phase 2, also in Canada. But because the technology was ready, platform, so they were very rapid to move. And what I'm showing here, this appeared May 22nd. It's the result of a phase 1 clinical trial in human in China with the adenovirus vector COVID-19 vaccine. This is an interesting site. Anyone interested in what's ongoing in the different vaccine under development? This is a very long list. There is 126 that are preclinical. Currently, there are 10 that are clinical, and among them, the top 2 are vector vaccine, and both are adenovirus. This one is the one run by University of Oxford and AstraZeneca, which is a chimpanzee adenovirus. In fact, it's a serotype that is different, and there is a reason there. So briefly, I will run over a very recent work that we have done. Okay, the Newcastle disease is the disease, is not a vector that we are considering, but yet, I don't want to mix you too much by bringing too many viruses, but that's the nature of the field, actually. We take advantage of the virus, and surprisingly, even the Newcastle disease virus that is currently a disease for dominantly poultry, as we will be discussing, has been identified for different use, in fact, has been also and is currently evaluated as an oncolytic virus. Okay, one form is considered as, but we are more or less in the same field. This is a therapeutic vaccine. We are in the space of oncolytic virus. So in our case, what we have done is apply, but the adenovirus technology, by using the surface protein of the Newcastle disease virus, and that was a project funded by the International Development Research Center from Canada. I think funds were also coming from the Gates. The key point is that the project was geared toward protecting the source of living of countries in East Africa, and the collaboration was dominantly with Ethiopia. As you will see, we collaborated also with Tunisia, and the critical point was to design a vaccine, recombinant one, based on the principle that I described, and we started by sequence analysis of what was circulating, confirmed what would be the most efficient construct. So we generated the adenovirus construct, and more than one, in fact, because we had not originally good idea of what would be protective as the antigen. We had to optimize, obviously, the making of the adenovirus, but because of the primary experience, we were rapidly to bioreactor production, and we generated sufficient material with significant number of constructs. Up front, this is part of what we call the vector design, of the antigen design. You don't know exactly what will make the critical difference. And in fact, we looked at different promoters, chicken beta-actin promoter versus CMV. This is more or less the basic that is used in many of the vaccine, and different combination of F protein and hemagglutinin neuraminidase that are associated with the NDV, Newcastle disease virus. And obviously, we had to do all the work that was required. There has been an evolution on medium, so we looked at the medium first in the growth phase, but interestingly, we were able very rapidly to generate this at cell densities that were reasonable without losing dramatically the yields. I don't want to bother you more. You get the point that we need to do extensive process development, in fact, especially in this case, that to address animal vaccination, cost effectiveness is critical, and you have to manage to make your vaccine safe, efficient, but also cost effective. And clearly this is one of the challenges also that we will run with any vaccine, because even for human, we're talking about global use and at price that are accessible to anyone. So we started with the batch mode, which was essentially to generate sufficient material for animal studies to demonstrate efficacy and immunogenicity first, and protection. Production in bioreactor, fully documented. People very highly trained to the engineering aspect, because this has to be documented. If you are gearing toward a process that would be going to industrial application, you have to start the work in the process development as early as possible. And this is the animal study that were carried on in Ethiopia, the National Veterinary Institute. And the key finding is essentially here. We had full protection with the adenovirus expressing the F protein. In fact, it was a surprise for us, because we were expecting more protection with the F protein, with the hemagglutinin neuraminidase that were expressed at higher density on the Newcastle disease virus. But the F protein was the protein that was allowing the fusion of the virus, and turned out to be the dominant antigen. So these are very exciting results. It's not yet published, it's under review, but which will lead to developing a vaccine that will be used, a recombinant one, for poultry protection. So in brief, adenoviral vector production completed at pilot scale, satisfactory yield, sequential batch and perfusion mode. We had to work actively on understanding the kinetics, and yet we believe that there is yet a lot to learn there. The overall process with the example that I showed was scaled up to the 500 liter, and that was limited by the capacity that was available. Otherwise, it's still a level 2 operation. And you might be surprised, but there is very few places actually that are equipped to operate a system with biosafety level 2 requirements, even less when it's biosafety level 3. I'm looking to the time, so I will run over the remaining example. I think you are well aware, because it's highly related to what's happening actually. There has been a warning with the Ebola virus outbreak in 2014, 2015. The Ebola virus is a fatal infection. The virus was kind of known since the 1976, and the death rate, the case fatality, are 60 to 90%. So this is the disease, and obviously the people realize that there is a need to develop a vaccine and post-exposure treatment, referring to the fact that the vaccine might not be fully active post-exposure, so you had to protect with monoclonal antibody. I just want you to get back to the dates actually. The crisis, the emergence, was publicized in 2014. And this is when WHO announced that this is of global concern. It wasn't a pandemic, it was outbreaks, well localized, but look at the date actually. There has been work on different approach for protection since the 2003, 2007, 9, 5. We were involved 2005 with this VSV, but also with the adenovirus quite early. preparedness, in fact, behind the scene. And the reality, to be fair, that most of this research were funded by biodefense. The fear at that time was the Ebola could be used as a biothreat. And we benefited from that funding also. So in short, I will tell you the story of the VSV vaccine, and I will stop just after this. Application of the RSV, replicative vesicular stomatitis virus, has been developed originally at the Public Health Agency of Canada, was licensed to a small company in the US, New Link. Clinical lot have been made in the 2013, and that was a good idea because some doses were available actually to be tested right away when the crisis get to the high. Many organizations were involved, PHAC, BARDA from the Human Health Services, WHO, etc. Turned out that Merck decided for probably not a commercial reason, but for what we can call recognition reason, acquired in fact the rights and started manufacturing this vaccine. These are the site in Canada, in Winnipeg. This is where the work started and what has been developed. In a nutshell, the VSV is also a vector, one of preferred vector. It's an enveloped virus. So the strategy that is used is replacing the G, the glycoprotein on the surface of the VSV, by the glycoprotein of the infectious agent that you are addressing as a vaccine. In this case, it was the Ebola, the glycoprotein, or the Zaire Ebola. That's why we refer to as ZEBOV-GP, glycoprotein. And very rapidly, there has been demonstration, in fact, of efficacy that has been published quite rapidly in 2015. Once again, because the product was already developed, was ready, and it went to clinical manufacturing. This is in a nutshell the process as we run it in our lab. Cell expansion, cell seeding, virus infection, virus harvest, purification, and we hope this is not yet achieved, and this is a problem. In fact, although it's successful, this vaccine is still delivered at -65 or -60, which make it quite difficult to distribute in remote area in Western Africa. And this is the reason it's very labile, actually. It's no more, it's a live virus. So it's losing all its infectivity at temperature 42 within 1 day. So you want to protect it. That's the reason behind. One interesting observation, this is referring to the quantification that we always try to put in place. Actually, this is the accumulation over a run of production of the VSV Ebola glycoprotein produced in viral cells. A digital drop PCR represents the total quantity of genomic material associated with each vector, whereas the infectivity. So although we see a drop in the infectivity, in fact, the total particles are still there, are maintained. So for a vaccine, it's very important to realize that we have a load, which is total particle. And in most of the case, I mean, the dosing is calculated based on the TCID or infectious unit, if it's a live virus. So this is something that we identify. I think I will stop here. There is a number of other vaccines and that are based on the principle of, but I just want to conclude by saying that our job in many cases is to really practice the science to achieve something that is beneficial. And I like this quote from Cesar Milstein that until it benefits equally by the really poor of the world, we haven't achieved much. And this applies extremely well in the case of the vaccine. We will find a vaccine, but it has to be accessible to all. And this is why finding ways to improve the safety, the protection, obviously, but also the cost effectiveness is absolutely critical in the space of vaccination. Thanks.
Thank you very much, Dr. Amine. I just have a few questions to ask you. One is, are you having any tie-up or technology transfer that you've done with the company? Are you working alongside a company for your vaccine development, or is it just like medical university by itself?
It's many has been transferred. What I described, the two cases have been essentially transferred actually. But you see that the companies are interested when things are ripe. You have to do, this is an area that is often neglected because it's requiring, it's extremely costly to manage. For me, it was possible when I was at NRC. It's challenging doing it in an academic setup, okay? But I would say that 50% of our activity is transferred because that's a purpose actually. This is a sign of developing something successful if it's transferred. If it's not transferred, it's going to sit there and really not being used.
Okay.
I'm responding to your question.
Yeah. And I have another question. Like, I mean, this has always been skeptical. Like the vaccine research has always been skeptical. Very, what do you say? Like, it's like a false sense of optimism at times when, like vaccines for polio and those kind of things were actually established and were working. But at the same time, we have like bad examples of HIV as well. When Merck launched the V520, the weekend adenovirus with the HIV genes, like, it was a big kind of breakthrough when suddenly within a few weeks they said the clinical phase 2 didn't go through properly. So from like, I mean, it's very difficult to actually predict, especially with viruses. So in this scenario, how do you think that this area of research will prosper, or how confident are you in vaccine research?
I think this is an important question that you are raising, actually. And my only response, I'm those who believes, I refer to 10 in clinical trial, okay, vaccines listed by the WHO, and surprisingly, some are already in phase 3 with the adenovirus chimpanzee. Phase 2 with the other adenovirus. There is a number of messenger RNA, there is recombinant protein, VLPs. There is obviously, I was surprised to see, although it's a level 3, there is inactivated vaccine that is also moving forward for clinical trial. A lot, there is 126 that are in preclinical, okay? And in fact, in my case, we are involved in 3 types of vaccines under development. Subunits, we are working, I can tell you, on the receptor binding domain to be used with an adjuvant and hopefully to be delivered nasal. We are working on the adenovirus with the RBD, differently from the one that I referred to from CanSino, that is working on the whole S protein. And we are working on the VSV. This is the indication that no one can predict. And you are absolutely right, actually. But at this time where the people are expecting, I'm worried, actually, that the message might be too positive to fool in a way the people, because you are right. After 30 years of investment, there is no HIV vaccine. And with the trial we refer to with the adenovirus, in fact, revealed something that has nothing to do with the adenovirus, revealed our and complete knowledge on the immunology response actually, and how this can be driven by some system. It was an activation of T cells, and that was the best route for the HIV to enter. So that was a learning, in fact. But so I think there is evidence that in some case it did a good job in the situation of Ebola. I showed some example where there is a number of applications with animal vaccine. I'm referring to the adenovirus. The VSV has one good example, is once again the Ebola. What we are doing here is essentially trying to mimic nature, actually, because the immune response is sufficiently broad, but the challenge is what is everyone currently developing a COVID-19 vaccine is worried about the cytokines store and trying to do everything possible to not get in that area of overactivating the immune system and turning the immune system against the vaccinated individual. This is not something that is neglected. So in summary, no one can predict at this point. You are absolutely right. And that's why, I mean, the support should be provided to all the possibilities, and hopefully among all those possibilities, one will win with the appropriate attribute, safety as a primary, efficacy certainly, and cost effectiveness.
Okay, thank you so much.
My pleasure.
I have a couple of questions from the audience.
Yes,
One
please do.
by Abhiram. She asks, since the VSV vaccine for Ebola is less stable at higher temperatures, why can't we go for lyophilization process as in monoclonal antibody formulations? With suitable supporting excipients, a lyophilized vaccine formulation can withstand high temperatures or not? Is the question.
Thank you. I proposed this project to my funding organization last year, and they rejected it. It's not sufficiently scientific, they said. But the value is such that in my case, I feel that it's so important. You are right, actually. And we know that enveloped virus can be lyophilized, and it's a high value to achieve that. Why it hasn't been done? Because of the so critical aspect of the clinical trial and changing something in the process, which is major, in fact. At the point is very difficult to introduce. So for the next generation of Ebola vaccine, there is no doubt that it should be lyophilized. But if it was taken into consideration at the early stage, that would have been acquired. But the history shows that it was done in a crisis time. In reality, Merck, when they took over for the vaccine, they changed zero. Okay, they are still producing in roller bottles. That doesn't mean that Merck doesn't have the abilities to do it more efficiently, but because it has been approved on a process that was already validated, although it was not the best process, it's staying there, and there is room definitely for improvement, and I would love solving that problem, in fact.
Thank you. The next question is anti-vector immunity due to vaccination or pre-existing immunity will be a problem in humans?
I didn't get it well, the question.
Is anti-vector immunity due to vaccination? Is it due to the vaccination or pre-existing immunity? Is it a problem to humans?
For the adenovirus, that was a critical point when used for gene delivery, because there has been a rejection, obviously, of the vector, and that's eventually the reason why it was, for some application, successful, because it's inducing, broad immunity. This is, I'm tempted to, and some will make the jump, actually consider the vector as the adjuvant, essentially. Okay, you are providing the antigen that might be expressed or exposed, but something else that will trigger a broader cellular response. And that's what is looked at. Is it 100% controllable? That's yet to, so in case of, people are looking to different scenario. If, for example, we boost with another vector, there is some consideration. It's the immunity of the vector is an advantage in the case of vaccination, whereas it's counterproductive if you want to deliver a transgene, and that's in gene therapy in general. It has been abundant, with the exception of the cell therapy aspect when it's targeting the cancer cells. So I hope that I responded to your question.
Yeah, thank you, sir. And I have a couple more questions. One is, in case of virus, huge antigenic drift is seen. So how did you manage to overcome this?
A huge, sorry, could you repeat the question?
In case of viruses, huge antigenic drift is seen usually. So how do you manage to overcome this?
But we want it in terms of immune response in some case. It's the balance that is looked at. In fact, you see, vaccines are a balance between safety and really activating the immune system to get sufficient efficacy in it. And that balance, if you take, I mean, a subunit vaccine, here we will be taking the RBD, receptor binding domain of the S protein, trying to minimize any chance to get in areas to induce cytokine storm, etc. But we know upfront that this will not be an effective vaccine. And upfront, we know that it has to be adjuvanted, and the role of the adjuvant is bringing the other element of the immune response, which might induce cellular response and long-term, hopefully, protection. So it's an area of extensive debate because, I mean, I think we are dealing with the complexity of the immune system, and I'm not at all an expert in that area, actually, by what I showed you. But there is a number. This is why, I mean, you can cure a number or control a number of disease in mice, but until you get to human demonstration, it's nothing is done, and that's one of the reasons actually that that we need to go with a different platform, with a different form, to achieve something that will certainly make the difference. But we hope with developing in parallel such a number of that we are creating capacity for the future, actually.
Thank you. And the next question, I think, is virus has many subunit structures. So will we be able to drift their antigenic property? Why can't we assume all subunit serotypes and prepare a cocktail vaccine against them?
That's surprising. That's a reason what emerged is what we call the dominant antigen. Okay, so if you want to, for example, one of the vaccine that is well known is the hemagglutinin, and we use just the influenza, and the influenza, obviously there is a number of protein on the surface, hemagglutinin. Neuraminidase is playing a critical role. So it turned out that it might enhance, I mean, the response, but it's not currently considered in the formulation. The recombinant vaccine that have been created focus just on the HA. I was surprised by the data I have shown you with the Newcastle disease virus, that we went for the different, for the most exposed element, actually, the hemagglutinin and neuraminidase. And in that case, we didn't have protection. Whereas with the F that was less represented on the surface, we get a protective response. And that result was kind of repeated. And there is a consensus in the field, actually, with many other viruses. With the RSV, for example, it's the F protein that is dominant. So we go to this notion of dominant versus, but you can mix, and there is strategies where, I mean, there is people that are going with the matrix and the membrane protein of the COVID-19, additionally to nuclear protein too, will be considered. Just because of our lack of knowledge. You see, we see a virus entering, and there is nice models to predict how this is happening, etc. But we are knowing better and better on a daily basis on the COVID-19, actually. I know these are long responses, but your questions are extremely important. But unfortunately, there is not a simple answer to what you are asking, actually.
Okay, thank you very much, Dr. Amine. It's been a great pleasure to host you. And I'm really extremely grateful to you for spending your valuable time with us. I always thought experts like you will benefit the students and the faculty, especially in India. So I once again thank you for your presence and for your wonderful lecture. Looking forward to India soon.
My pleasure, and happy to be around, although remotely. I would have loved to see many of you, but maybe we'll have some occasion in the future. So take care and keep safe, and the field is in need of people in the space of vaccine bioprocessing and manufacturing. So it's an important area to cover, actually.
Okay. Thank you, sir. Thank you so much.
My pleasure.
Thank you. Bye.
Okay. So I'm leaving you good continuation of your...
Get in touch, sir. Thank